defects. They implanted the material, followed by they examined by micro-CT, histological staining and immunofluorescence staining. They evaluated how much the new bone is formed and macrophage phenotypes after 8 weeks. So they statistical analysis, they used the student t-test for comparison between two groups. The one-way ANOVA for comparison between multiple groups. The Turkey's post-hoc test for performed after ANOVA to identify specific groups differ significantly. The p-value was set for greater than 0.05, which was considered statistically significant in these tests. Then coming to the results, so the material characterization, the material showed uniform spherical morphology and high porosity and stable structure. FTIR XRD confirmed successful incorporation of cerium and irisin without phase separation. The ion release studies showed sustained cerium and silicon release, beneficial for antioxidation and osteogenesis. The cytocompatibility, both materials were tested to be non-toxic and macrophage and the stem cells. Two macrophage and stem cells showing excellent cell viability. The antioxidant properties, the material reduced the reactive oxygen species accumulation effectively, demonstrating powerful antioxidant activities via cerium redox cycling. The immunomodulatory effect, the material shows reduced inflammatory cytokines and increased IL-10 and argin-1 cytokines. The flow cytometry and immunofluorescence confirmed the M1 to M2 macrophage polarization, improving anti-inflammatory and healing responses. The osteogenic differentiation, the material showed enhanced ALP activity and mineral deposition compared with the material in their individual parts. The upregulation of osteogenic genes was confirmed to improve bone differentiation, and the molecular mechanism, the Western blotting indicated activation of pathways and which are crucial for M2 polarization and osteogenic signaling. So blocking these pathways inhibited the observed effects, confirming the causality. The in vivo bone regeneration, micro-CT which they did, showed substantial new bone volume and the material treated defects compared to the other groups. The histology revealed well organized bone tissues and reduced inflammation. Immunofluorescence confirmed more M2 macrophages and higher osteocalcinin expression in the group with the material injected, and overall outcome, the material showed a synergistic effect. Early immune modulation through M2 polarization and late stage promotion of osteogenesis, leading to superior bone healing. So coming to discussion, this study designed an irisin loaded cerium bioactive glass that synchronizes immunomodulation and osteogenesis through a dual pathways, providing a promising platform for next generation bone repair materials and that can heal bone defects more effectively and naturally. The strength of this, they developed a dual function biomaterial as said before. So to control inflammation and promote bone regeneration, they combined natural molecules and therapeutic ions for complementary effects. They demonstrated clear molecular mechanisms. Also, they included comprehensive in vitro and in vivo validation, strengthening the translational potential, and also showed sustained release behavior, ensuring long-term bioactivity. The limitations, the precise long-term biodegradation of the material was not extensively studied. The dose optimization of irisin-loaded and release kinetics need refinement for clinical scaling. The human cell models and large animal validation are still required before translation. The study mainly focuses on macrophage, that is osteoblast, crosstalk and other immune cells. Basically, other immune cells were not evaluated in this study. So the study design, this overall adhered to the arrived guidelines about 80 to 85%. The guidelines which were not followed. Randomization, the study did not specify whether animals were randomly assigned to the experimental groups. Blinding, there was no explicit statement about blinding during sample processing histological analysis. The article also lists the number of animals to be used but provides no statistical or power analysis. The inclusion and exclusion criteria were not mentioned as per, and the protocol registration was there was no indication that the study protocol was pre-registered before experiment. The welfare and monitoring details, while they had the ethical approval, the welfare and monitoring details were not mentioned clearly. So that's all for the article. Thank you.